Okay, hello everyone. Before we start with today's lecture, I wanted to remind you that you have your weekly quiz that's due tomorrow, Thursday night, by 11:55 p.m. So hopefully this is not a surprise to you, and that you've already had a chance to take the practice quiz, which is up there. Take the practice quiz just so you can run through and see what the questions are like, and make sure that your Laulima is working for being able to take quizzes. And then make sure you take your first real quiz that's due Thursday night, 11:55 p.m. Okay? All right, good. So it's important that you get into that routine because we have a quiz pretty much every week of the semester. The class schedule file that's available in Laulima indicates which weeks we have quizzes and which chapters those quizzes cover. So you always have the ability to know what you should be reviewing. All right. Okay, good. So today, let's continue talking about the innate immune system and the part of the innate immune system that needs to immediately respond to a threat that enters our body's tissues, and what types of proteins and cells are part of that response. So you will recall that one of the most important proteins involved in our immediate innate response are these complement proteins, in particular, complement protein C3. Last time we began to look at how it is that this protein that's made by the liver and is floating around in our blood, these complement proteins, get activated so that it can do processes like, for example, attaching to the surface of pathogens like a bacteria or a virus particle, thereby making that pathogen much easier for something like one of our macrophages to stick to, sort of like Velcro, right? And then being able to then make it much more efficient for macrophages to go ahead and phagocytose or eat those bacteria and thereby destroying them. So recall that we said that there are three main pathways for activating complement proteins. The one that we have been looking at so far is the alternative pathway. It's the first one to react. So it involves complement proteins that are floating around in our blood and our tissue. And remember, it has to do with the way that the complement protein is activated, cleaved, so that it's able to form very strong covalent bonds to other target molecules, for example, on the surface of a bacteria. And then once that complement protein is attached, it can then facilitate the very rapid spreading of more complement proteins on the surface, thereby quickly opsonizing or covering the surface of a pathogen, and alerting our immune system that it's now easier to go ahead and destroy. We will also talk about the lectin pathway and the classical pathway as our semester goes on. But for today, I wanted to start off with just a quick review about protein structure. I think most of you have already taken Bio 275, where we spend quite a bit of time understanding how it is that the amino acid sequence of a protein influences the three-dimensional shape of a protein, and how it really is all about the three-dimensional shape of a protein that determines how it interacts with other sort of partners, molecular partners, like other proteins. Right? But I do want to stress that it's important that all of us are on the same page, and that if there are some of you who have not taken intro to cell biology yet and aren't really sure about this whole, like, I don't understand what you're talking about, proteins and stuff, let's just quickly do a review to get us all on the same page. We said that in the book, oftentimes the illustrations are oversimplified, like the complement protein C3 is just this light blue colored egg, right? And there's like a pair of scissors that come along. But we want to be careful, because while we want to understand the concepts behind it, we don't want to get too wedded to just thinking that complement C3 looks like this. So let's go ahead and talk about different ways that we can visualize or imagine the structure of complement protein C3 to help us have a better kind of foundation for being able to talk about things like how receptors and proteins work together. These are 4 different ways of thinking about the structure of something like complement C3. The first is the way that the book tends to present it. So in the upper left-hand corner here is sort of a very reduced, functional, simple geometric kind of shape representing C3, right? That's the blue oval. And we said that within the structure of this blue oval, there are these side chains of amino acids that present the possibility of forming a thioester bond. And that if you activate this complement C3 and it becomes complement C3b, and the other little part of it is C3a that gets cut off, it floats away, then this thioester bond is exposed. And if this is happening right near the surface of, for example, a pathogen, right, then some molecule on the surface of that pathogen that we'll just generically call R, right, some sort of group that this thioester bond can interact with, you can form a very strong covalent bond that then begins the process of the opsonization or the coating of the pathogen to spread. But let's look at different ways of visualizing this C3. At its most fundamental, this C3 protein is a long chain of amino acid residues that are linked together by peptide bonds, right? So it's a polypeptide chain. One way we can think about it is if we were to unravel the C3 and kind of make it straightened out, we can map out the different amino acids that are at different positions within the structure. So this is the first 100 amino acids that make up the complement C3. And you can see, again, if you've taken intro to cell biology, you recognize that these are the single letter abbreviations of amino acids: methionine, glycine, proline, threonine, serine, etc. Right? The first 100 or so amino acids are actually highly conserved throughout evolution, meaning that this sequence of amino acids, because it is very useful in forming a particular shape, is something that we evolved to use as kind of like a theme that appears in other proteins that have related function as well. Right? These are, of course, coded for by our genes. So we have a gene that encodes for this amino acid sequence as part of encoding for the production of C3 protein. But this genetic sequence can also be found in other genes of other immune-related proteins that might also take advantage of having this similar structure as part of its final protein shape. So this first 100 or so amino acids, we call this a macroglobulin module, right? Because we see this used in other proteins of the immune system as well. I always say, when Mother Nature comes across something through evolution that works really well, it's not surprising that if you fast forward hundreds of million years through evolution, that things that work really well, right, you see them again used for different functions. So this macroglobulin domain, a C3 protein, if you were to lay it all out as a straight polypeptide chain, we might say this first part of it consists of these, 5.5 macroglobulin modules, right? And then you can see the different colors here represent other functional regions of this protein that really only make sense as functional when the protein is folded up in its correct three-dimensional shape. But if you unravel it now, at least you can identify what amino acids are part of this structure. For example, this light blue region here, labeled ANA, right, is the C3A region of the protein. In other words, if we were to look back at the simplified cartoon up here, this would be the stretch, the small polypeptide stretch that gets cut off and floats away as C3A. So you can see in the complete protein, it's right in the middle over here. Right? Over here, this TED domain is the thioester bond containing domain. And this little orange dot is the thioester bond, right? So that means this up here, what you see here is part of this TED domain of the protein. And then you have the C-terminal end of the protein in pink here, right? This eventually ends at the carboxyl terminus of the protein. So this is the C-terminal end, compared to way at the start is the amine terminal end of the protein. Okay. So that is one useful way of looking at proteins, because it provides sort of a nice laid out map of where these different things are in the amino acid sequence, right? But of course, right, having it just as a straight line is not very realistic. The reality is it's all folded up. So that brings us to our third way of thinking about what this protein looks like. If you were to take that map that's down here, the primary sequence, let's say, I mean, it kind of looks like a choo-choo train or something like that, right? But we say in reality, it's not straight out like a choo-choo train like this. It's all folded up on each other. This is how the domains fold up. And you can see here are the green macroglobulin domains. Here is the TED domain with the thioester bond. Here's the little ANA domain that becomes the C3A that gets cut off. Right? Notice this way of representing it now proves to be much more useful for understanding how the C3 structure is changing once it gets activated and can bind to a target. So here is C3 in its inactive form. So in other words, it still has the C3A part attached to it, and the thioester bond, the little orange triangle here, is hidden within the protein, right? So that's like this on the left over here. Once the protein is cleaved and this ANA part gets cut off and floats away as C3A, what happens is the rest of the protein is now unstable. Right? The way it was folded up before, it was happily folded up that way because all the little side chains of the amino acids, this is how they happily kind of found their own little space. And this whole thing is surrounded by water, right? But now you mess with that by removing part of it, the whole thing has to shift around a bit to get into an alternative conformation that once again is kind of comfortable for all of the bonds and things to be in. So the protein, or the gene that codes for the protein, evolved with this very particular amino acid sequence, so that when you go ahead and cut off this C3A part, the ANA part, and the resulting rest of the protein has to shift around, look what happens, right? The thioester bond containing domain now flips down and points down, right? So that's why the thioester bond now, just like in the cartoon on the left, is available to bind something. I think that's really cool when you realize, yeah, I get it now. This is what's really kind of going on here. And it's all based upon the structure of the protein, trying to find a conformation that is energetically stable and favorable under different conditions. Now we can take this a step further. We can say, yeah, but we're still representing this as like ovals, triangles, and rectangles, right? What does it really look like? If you were to really look at the peptide backbone and follow it through this molecule, this is what you get. Right? So this is now the C3 before it's activated. The light blue over here is the C3A that's still part of it. And you can see the thioester bond side chain is tucked inside. Once it's activated, the light blue part has fallen off and it's floated away. And the whole thing has to shuffle around a little bit. And in the process of doing so, the thioester bond is now pointing down and it can now react with something. Right. The final way of visualizing this, which is my favorite way of visualizing it because I think is the most useful, is looking at it like this, right? So now instead of just following the polypeptide chain around like some sort of maze or some sort of like little game that you play on the placemat at Zippy's or something like that. Now, if you actually fill in all of the electron orbitals of all of the atoms involved in these amino acids and the side chains, this is now what you get. Right? So this is the inactive C3. This red part is the ANA or the C3A part. Once that gets cut off and it floats away and you're left with C3b, you can see it all shifts around. And then this is where the thioester bond is now available to interact. Okay. So that's my sort of crash course in protein structure. Hopefully for most of you, that's kind of a review, but it's a nice reminder, right? That if we are going to talk about through this class about how different proteins act as signaling molecules and other proteins receive, those signaling molecules, being able to really in our minds imagine that these are three-dimensional things makes a lot more sense, and it will help you in being able to really conceptually understand many of the concepts in this class. Okay. Okay. So let's continue talking about how is it that complement proteins like C3, right, can both indirectly lead to the destruction of something like a bacteria, indirectly meaning they label it, right, through this process of opsonization, but ultimately it's up to something like a macrophage to come along and eat it. Right? Not only can they work that way, but complement proteins can also directly destroy targets. How? Through another chain of mechanisms that lead to the destruction or disruption of the membrane of the bacteria. So previously, we had looked at these top two panels here. We said that once you get a C3b that has attached to the surface of a pathogen, and that C3b can recruit another complement protein, factor B, that then gets activated, so it becomes BB because the little N cuts off, right? We said that on the one hand, if you follow this arrow here, this is now the sort of chain reaction that we've talked about last time. That C3b BB, which we call a C3 convertase complex, goes to work converting or activating other C3 molecules that are floating by. So this C3 molecule comes, and this C3b BB is the right three-dimensional shape, right? Think about these previous slides that are showing you. It's the right three-dimensional shape so that a C3 molecule comes and it fits into it nicely. And it fits in exactly the right position so that this yellow BB part, because it's an enzymatic or it's a protease, it wants to cleave other proteins, it will go ahead and act as the scissors that then activate this new C3 molecule, causing its C3A to float away, and that C3b molecule then gets stuck to the surface. And we said, importantly, once the C3 is activated and released, the remaining C3b and BB, well, it just keeps going around and around over here because it can recruit another C3 and continue sticking more C3bs on the surface. But also importantly, each one of these C3bs can themselves recruit one of those little yellow factor Bs, right? So each of these can become its own C3 convertase. And that's why the opsonization or the coating of the bacteria happens very fast, exponentially, once this process gets started. Alternatively, though, another thing can happen. There's going to be a brief moment when the convertase complex, the C3b and the BB, is still hanging on to this activated C3b, right? In other words, before the C3b has attached and stuck to the surface, there is a moment when this whole complex is still together. And that's what's shown down here, right? So let us pretend, right, that instead of up here, this moving on to this step over here, and then the convertase complex keep going this way, that there is a brief moment when that C3b is still hanging on. And in that brief moment, that combined shape together is the right shape for a complement C5 protein if it comes floating by at the right time to sit on top of it. Right? And if the C5 protein sits on top of it, then the little yellow BB, the protease activity, will end up cleaving the C5. And surprise, surprise, it gets fallen apart into a small C5a that floats away and a larger C5b that floats away. All right. So what does that mean? That means at any given moment, right, starting from here, you can go in this direction or you can go in this direction. But keep in mind that both of these happen very rapidly, right? So after either of these is done, you have the freed up C3b BB again, right? And importantly, keep in mind that both of these are actually happening simultaneously because, oops, sorry, even though the cartoon we're focused on only one of these C3b BB complexes stuck to the surface. Of course, once you have a ton of these on there, right, then both of these processes are happening at the same time, right? with each given one. So both of these pathways are happening simultaneously. So what is this C5 pathway all about? That has to do with this. If you think about something like a bacteria as really just being like a sack of water or a baggie of water, right? Because that's what it mainly is. It's like 70% water with, some genetic material, proteins, carbohydrates, some lipids in there, right? A very effective way of destroying this bacteria, other than actually phagocytosing it, like a macrophage at phagocytosis, is you can poke holes in it, right? So if you poke holes in it, then the balance between the osmotic pressure inside versus outside gets really messed up, and the bacteria can't manage things. The water would probably rush in and cause the bacteria to explode, right? Just like what you see here. Right? So that's what this C5 pathway is going to do. When you convert the C5 into C5a and C5b, this C5b floats off and starts what we call the terminal complement cascade. Ultimately, what it will do is it'll start recruiting other complement proteins C6, 7, 8, and 9, to form a special complex on the surface of the bacteria known as a membrane attack complex, or MAC. Okay. That's what's pictured here. The MAC is essentially poking a hole in the surface of the bacteria. It is a complex that makes a tunnel through the membrane of the bacteria so that water now flows uncontrollably through it. Here you can see this is the C5b that from the previous slide that was made, and it begins to recruit complement C6 and C7. So once those come together, so these are three separate little proteins now that are sticking together because they're the right shape to fit together, right? Once those three fit together, it sticks to the surface of the pathogen, and complement C8 is recruited, and C8 inserts through the membrane. And now probably the most important step is it recruits complement C9. Okay? Not just one molecule of C9, but it recruits a whole bunch of C9 that ends up being assembled into a ring. Okay? A ring that ultimately forms the MAC because it penetrates through the lipid bilayer. This on the right is an actual electron micrograph of these membrane attack complexes that were formed on the surface of a bacteria. So it basically turns it into Swiss cheese. Right? Once this happens, the bacteria just lyses, right? Because all the water gets completely unregulated, and it most likely is going to just fill up with water and burst. And so the destruction of the pathogen is very swift. So what do I want you to know from this, right? I don't really care if you could or could not draw, these figures coming together, because it's all a cartoon anyhow, right? I mean, it'd be kind of silly for me to say, hey, you have to be able to draw this, when in real life, this is just arbitrarily how this particular illustration draws it, right? But I want you to know is that the C5b comes from the C3 activation pathway, right? Because through making C3 activated and C3b sticking to the surface, that's how you then are able to recruit factor B, and you can get more C3 activated that starts opsonizing, but at the same time, right, some of them are going to be making C5b, which then starts this process. So both are happening simultaneously. As the bacteria is getting coated with C3b, it's also being poked full of holes. Right? Very effective, as you can imagine. I'm never satisfied just with these simple geometric shapes. So let's look at a more accurate illustration of what these different complement proteins look like as they come together. Right? So that's what this is here. So you can see this is the C5. Right? And once it's activated and part of it gets cut off, it makes the C5b. So that's the greenish color structure here. But you can see because it's now the right shape, it recruits complement C6, which is the lighter blue. You can see very nicely in this illustration how they really fit together, right? Like one of those three-dimensional jigsaw puzzles, right? You can also see how once this shape forms, it recruits C7 and C8. So that's the kind of pink and purplish structures here. And ultimately C9, this kind of green structure here, gets recruited, and a whole bunch of C9 form a ring around here. Okay. So just be familiar with this membrane attack complex as another mechanism by how through which the complement C3 that first gets fixated on here can result in the direct disruption and destruction of the bacteria. Okay, and just be familiar with the concept that that involves the formation of C5b and then the recruitment of C6 through C9. Right? Okay, so hopefully, you think, that's really cool. And it's cool because these things are all mediated just by proteins, right? These little molecules that don't have any brain, they don't know what they're doing. Things just happen because they are there and they're the right shape at the right time so that they can come together and these things happen, right? So what's scary about that, like I told you last time, is, hey, what's to prevent this from happening to the surfaces of our normal healthy body cells, right? If we're making these different complement proteins and these different things happen when the complement proteins are in the right, combination and they start coming together, that could spell disaster for our body's tissues. We'd essentially self-destruct, right, through this mechanism. Just like last time, we saw examples of how certain proteins are present in our blood and on the surfaces of our normal body cells to help discourage complement C3 from opsonizing them, right? We also have certain proteins that help discourage the improper formation of these MAC holes. So an important one is this protein CD59, pictured here in orange. So CD59 is present on the surface of our body's healthy cells. It's also called protectin, right? Clever name. And the way it works is it gets in the way of the MAC complex that's forming, preventing it from actually finishing the formation by recruiting the C9. You can see here, that's what's represented here. So you have a membrane attack complex that's improperly forming on the surface of a human cell in your body, right? Again, it happens all the time because all it takes is the combination of these different complement proteins to be together, and it starts doing this chain reaction, right? But your human cell in your body has CD59 on its surface. Once this membrane attack complex starts forming, so it's made up of the C5b plus 6 plus 7 plus 8, it's the right shape for CD59, that's just kind of also floating around on the surface here, to kind of come and fit over it. And once it fits over it, it then blocks the recruitment of C9. So you cannot finish making the hole. All right. So this CD59, hey, it's coded for by your genes, right? Just like the rest of these complement proteins are coded for by your genes. Right? So you might think like, well, what happens then if you don't have CD59? You're in trouble, right? So people who are born with certain alleles or genetic defects in things like the gene that codes for CD59, right? They're more susceptible to all kinds of autoreactive types of immune reactions and things like that. And depending upon how impaired their version of CD59 is, right, it may actually lead to things like, it being fatal, a fatal condition. So maybe these certain people don't even grow up, right? They die in childhood. Another situation where CD59 comes into play is when we talk about cancer. So towards the end of the semester, we'll talk about cancer. But cancer involves cells of your own body mutating, right? Getting defects in certain genes that simply by chance happen to be changes in those genes that result in the production of different proteins or the complete loss of certain proteins that end up allowing a cancer cell to continue functioning. There absolutely are examples of certain cancers developing mutations where CD59 gets really ramped up in cancer cells. So in those cancer cells, you have a ton more CD59 on their surface. So when the immune system is trying to come and attack the cancer cell and destroy it, the cancer cell is protecting itself by having a lot of CD59 on its surface to dissuade or prevent the formation of these MAC complexes that your immune system is trying to do to destroy the cancer cell. Here's an example of a person who's born with some sort of deficiency, right, in CD59. So in their red blood cells, if we were to zoom in on the surface, here's the cell membrane. Here the CD59 is kind of drawn just as this funny sort of peptide chain. I know it looks quite a bit different than the orange cartoon over here, but again, it's a nice reminder, right? These are just cartoons, and depending upon who drew these cartoons, things can look really different, right? But the point is, people who are born deficient in CD59, the red blood cells are targeted by the membrane attack complex of their own immune system, resulting in destruction of large numbers of red blood cells. And this can lead to anemia, all right, weakness, fatigue, low oxygen levels in the blood. And again, when you say deficient, depending upon the severity of the deficiency, depending upon the nature of the mutation, it might just be that the CD59 doesn't work as well, or it might be that the CD59 really is not present at all, right? That can lead to varying amounts of disease, right? So that's the main way that our body protects against these, we call the MAC attack, right? So yeah, it's a MAC attack. Every semester I show this slide and I tell myself afterwards, I have to take this slide out because it's so dumb. But then I remind myself it took me like 2 hours to make it. So then I leave it in every semester. Okay, at least it helps you remember it. So far we've been talking about the immediate response of our innate immune system, right? So some of the things that we've talked about as part of this immediate response are macrophages are already there in your tissue, as shown here, right, patrolling around. They're like the mall cops, the very modest cost to maintain security system. If they bump into bacteria, they will phagocytose and destroy them. And we've started talking about other proteins that just function because they're the right shape and can also participate in the immediate innate reaction. So we've looked at complement proteins already. Let's go ahead and look at 2 other examples of immediate immune response proteins, macroglobulins and defensins. Okay. So macroglobulins are a class of proteins that were very early identified in the immune system. And that's why the particular amino acid sequence of different parts of them were seen used over and over again. And that's why these particular modules or different amino acid parts of them are called the macroglobulin domain, right? Because they were first identified in these macroglobulin proteins, but now we see them in other things like the complement proteins and things like that. So macroglobulins are immediate bacterial inhibitors. Okay. Bacteria, when it gets into your tissues, starts to condition your tissues to make it a better place for the bacteria to grow in. Right? It's kind of like when you move into a brand new dorm room, brand new apartment, brand new house, and it's all empty, right? Yay, it's your new place, but you don't really feel comfortable in it until you start doing stuff, right? You move your furniture in, you have all your books, your TV, whatever, stuff on the walls, right? To make it more comfortable for yourself. Bacteria is like that also. When it first gains entrance into your tissues, it starts secreting things to kind of condition the tissue around it to make it easier for the bacteria to multiply and spread and live. Right? Certain types of molecules secreted by the bacteria are proteases. So in other words, yeah, they are proteins that go around digesting or chewing up other proteins. Right? That's why infection by bacteria can lead to tissue damage. Right? That's why I don't think it surprises you that if you have a bacterial infection in parts of your tissue, right, you end up with potentially severe tissue damage as your tissue is being destroyed. And the bacteria is doing that through the use of things like proteases. So our immune system, our innate immune system, has evolved to have certain proteins to counteract that, to defend against many of these like proteases that are being released by the bacteria. We call these things protease inhibitors, right? And macroglobulins are an example of a protease inhibitor. This is the macroglobulin shown as this pink kind of, I don't know, kind of like a curved panel looking thing here, right? And then here is the bacterial protease represented as a green Pac-Man, right? So yeah, you know, heed my warning, right? If ever there is an example of the textbook kind of oversimplifying things just to get points across, this would be it, right? Because it's not really like a green Pac-Man being facing off with a pink curtain, right? But let's go with that. The structure of the macroglobulin has two important features in it. One is that it has a thioester bond, just like a complement protein, that's ready to form a strong covalent bond with some other target. But this thioester bond is currently hidden within the inactive macroglobulin. The other part of it that's important is this bait region. So if you imagine this pink curtain as actually a three-dimensional long peptide chain that you're following around somewhere, part of that chain is kind of presented over here as this bait, right? And it's in a position that's exposed to the outside so that it's quite possible that if a protease comes along, right, and wants to interact with a protein and cleave it and destroy it, that green Pac-Man will come and take the bait, right? And it cleaves this part of the peptide chain. When that happens, the whole protein, because of its structure again, needs to shift to get comfortable. This is the cool part, right? It is essentially hinged because the electrostatic nature of the structure, right, is kind of like suspended that way, just as long as the bait region is intact. If that bait region is broken, then the peptide chain collapses. And in this case, it collapses around the green Pac-Man protease. And in doing so, it allows the thioester bond to attach. And now this green Pac-Man is sequestered, right? It is now captured and neutralized, right? And it can no longer harm other proteins in your tissues. Hopefully, when you're looking at this, you're like, what? That's crazy. And you're like, wait, what does this thing really look like? Right? So of course, I'll show you what it really looks like, just as long as my screen sharing cooperates. Here it is, right? So down at the bottom here, this is the ribbon structure of the polypeptide chain of the macroglobulin, right? And you can see it's symmetrical. Right down the middle, if you put this dotted line, let's just look at the left half of it. This is the left half of it now. If you fill in the electron orbitals of all the amino acid side chains, and you can see here, this is where the thioester bond domain is currently held. And then this region across the middle here, which would be, I guess, this region over here, is the bait region, right? So when that bait region is broken, the whole thing closes up. Right. And I tend to think of it as like, it's like a bird of prey, like an eagle, right? Pouncing down on something and the wings kind of come around it, right? But it's amazing when you think this is real, right? This exists inside of your body. And all this is a protein. It is a chain of amino acids that through evolution, the order of those amino acids, right, forms this complex machine like a spring-loaded trap, right, in order to help protect you against bacterial secretions. Okay, another example of an immediate protein, immune protein, are defensins. Again, another nice name, right? Defensins. They immediately lyse pathogens. So whereas the macroglobulin sequesters and just neutralizes pathogens like that green Pac-Man in the previous slide, defensins are more actually about destroying the pathogen itself. And they do so, again, by poking holes in the surface of a bacteria or a virus. Defensins are very small. There are like 35 to 40 amino acids, so not very long at all. Right? And this is the structure of it. Both the yellow and the green, if you follow the ribbon around, I actually don't know which is the N terminal, which is the C terminal, but the point is you could follow it all the way around and it's one chain of 35 to 40 amino acids. But because of the nature of the amino acids, you have certain regions that have polar charged side chains off the amino acids and regions of this protein that are not charged and not polar. So as a result, you get an amphipathic molecule, right? One part of it in yellow is happy to interact with water, so that's the hydrophilic part. The other part in green is the hydrophobic part, right? Water molecules just kind of push it around, but it doesn't really interact. Actually, this illustration on the left is nice because it not only shows you the ribbon diagram, but very faintly you can see they superimpose the true three-dimensional shape once you fill in the electron clouds, right? So to me, this is a nice example of why visualizing it both ways is very useful. So these defensin molecules then, because of their amphipathic nature, naturally will insert themselves into the lipid bilayer, right, of cells like bacteria. So if these things are our phospholipid bilayer of the membrane of something like a bacteria, right, the defensin molecules, because they are amphipathic, right, the green part of them, the hydrophobic part, will naturally be much happier when the water molecules surrounding this shove them down into the hydrophobic interior of the membrane, right? Whereas the yellow part, the hydrophilic part, because it's charged, is very happy still facing out, where the water molecules out here are happily interacting with it. When a couple of these insert into the membrane close to each other, again, because they are amphipathic, they will also be attracted to each other, and they will essentially pinch off parts of the membrane like this. So here's 4 of them that together have pinched off parts of the membrane, resulting now in a hole in the membrane surface, right? And as a result, just like with the membrane attack complexes, with the complement proteins, the water rushes in and the bacteria is destroyed. So you might say, wait a minute, right? This is a lot simpler than that whole C5 membrane attack complex thing that had C5, C6, C7, 8, 9 forming this big thing. Why don't we just rely upon this? Right? The reason we don't rely upon this is if you think about it, because this is way simpler than the membrane attack complex mediated by complement proteins, this is only just one molecule, one defensin molecule that's coming down and several of them binding and forming this gap. This is a bit more dangerous because you have less opportunity to control this, right? So if this were actually present inside of our blood and started doing this to all of our cells, right, you'd be in trouble really fast. So as a result, we evolved that this particular type of mechanism of defensins involve proteins that are only expressed or secreted in things like our sweat or tears or into the lumen of our gut, right? Or inside of macrophages, the phagosome vesicles. Right? In other words, our immune system is very careful about where these defensin molecules are made, and they tend to be made in places that we don't worry so much about our body's cells being exposed to them. Right? So that's why, like in sweat on the outer surface of your skin, right, it's a great natural antibacterial. So defensins in your sweat keep the bacteria population on the surface of your skin under control, because your skin itself is just all dead keratinized layers of, dead skin cells, right? So you're not worried about defensins harming your own skin there. Likewise, the lumen of the gut, right, where your food is passing through, we've already studied this, that is technically outside of your body, right? So again, the cells of your body are not going to be in direct contact with this, right? But it's another nice example of how you can have a very effective antimicrobial mechanism that's mediated simply by proteins, right? Something that does not even involve another cell having to come along and attack it, right? So those are examples then of immediate responses, stuff that's always present, ready to go, okay? But remember, it is possible that the amount of bacteria that got into the cut, right, is more than what can be conveniently handled by something like macrophages or even the complement proteins there. Therefore, the second part of our innate built-in pre-programmed immune response is the induced response. So this really is when the macrophages are calling for backup to help out, right? They're not calling for the SWAT team yet, right? They're not calling for the adaptive immune response, because that's the super expensive overkill response, right? For now, they just need to get a little help from their other immediate effector cells. So that's our induced response. So macrophages are releasing cytokine messages. These are the orange triangles, right? As they're eating bacteria or as they are detecting different components of bacteria. And these cytokine messages then call for the start of inflammation, which is a very important process that we will talk a lot about. So I'm just going to mention that now, right? You get inflammation in your skin. That's part of this induced response. It also helps recruit other cells like more monocytes and neutrophils to come and help out. And as a result of a lot of this immediate activity going on, you're going to have a buildup of complement C3a and C5a pieces that are getting released by all the complement proteins that are activating. So these C3a and C5 pieces contribute to this induced response. So as a reminder, right, when these complement proteins are cleaved, you get the little C3a part that falls off of them, and the other C3b part is what sticks and starts opsonization, or likewise the C5, when it's activated, the C5b part goes off to form the membrane attack complex. The little C5a part that's cut off floats away. So these are what we call anaphylatoxins. That's why in an earlier slide, when I was talking about protein structure, they were labeled as ANA. It's short for anaphylatoxin. Right? Anaphylatoxin, you might have heard of anaphylactic shock, right? That's like when somebody who's allergic to bee stings gets stung. What it refers to is a systemic body-wide reaction to an immune response. Right? These little pieces of C3a and C5a, they bind to receptor proteins that are on immune cells that can interpret them and use them as a message to come help out. But importantly, they can also bind to the surfaces of immune cells that release things like histamines that cause a lot of inflammation to happen. And they can bind to receptors on endothelial cells, which are the cells that line the lining, or they are the lining of your blood vessels. And that causes the smooth muscle around your blood vessels to constrict. And that's all part of the potentially severe reaction if a person is dramatically allergic to something like bee stings, right? That's the anaphylactic shock, because the dramatic decrease in blood flow throughout their body can lead to things like them passing out and actually going into a shock that, if not treated, could be fatal. This is the last slide for today. This is just illustrating some of these things happening. So in this pink surrounding tissue are C3a and C5a that were released by complement proteins that are being activated. Here is a cross-section of a blood vessel that's running through here. So these purple things are the endothelial cells that line the blood vessel, and in the blood inside are all kinds of goodies, right? Other immune cells, other immune molecules like antibodies that we will learn about later. So the C3a and the C5a cause these capillaries to get leaky, and as a result, things like more complement proteins can rush out of the blood to help out where the bacteria is in the tissue, but it also allows things for monocytes, neutrophils, other leukocytes to come out as well and help out with the fight against the threat. All right, so these are all part of what we call the induced response. This is when the macrophages have called for help, and this is how components like C3a and C5a actually carry out the message for help. Okay, so let me stop there.